Good afternoon, and welcome to the 13th webinar in the Human Health Exposure Analysis Resource Program's Exposomics webinar series. My name is Jennifer Collins, and I'm a health specialist in the Exposure, Response, and Technology branch at the National Institute of Environmental Health Sciences and the Program Officer for the HERE Coordinating Center. This webinar series covers various topics of importance to exposomics. During today's webinar, Dr. Kurunthachalam Kannan will discuss new advances in the approaches and applications of quantitative biomonitoring for exposomics. Dr. Kannan is the Deputy Director of the Division of Environmental Health Sciences at the New York State Department of Health's Wadsworth Center. He is also a professor at the School of Public Health at the State University of New York at Albany. He has published over 900 research articles and is the top 5 most highly cited researcher in the field of ecology and environment. For over 30 years, his research has been focused on biomonitoring of human exposure to organic pollutants. Dr. Kannan's presentation will be followed by an opportunity for questions. As you think of questions, feel free to type them in the Q&A box, and I will read them to the speaker following his presentation. Alternatively, you may raise your hand and we will call on you to unmute and ask your question aloud. Dr. Kannan, the virtual podium is all yours.
Thank you, Jennifer. Good afternoon, everyone. I think I'm going to share my slides and see how it goes. You can see my slides and you can hear me, I guess. Okay, so good afternoon, everyone. Thank you for having me. Thank you for this opportunity to present at this webinar. And thank you all for joining this webinar. I'm going to talk about biomonitoring and exposome. My goal today is to try to convince you that biomonitoring is a fascinating research field in public health sciences. Often biomonitoring labs are more of used as service labs. Biomonitoring labs get samples from collaborators. We perform the analysis and submit the data back to our collaborators, and then we collaborate publishing the work. But biomonitoring is much more than that. So I'm going to talk about how we can enhance the biomonitoring program. So I will start with some of the definitions. You might have already heard of this many times. What is biomonitoring? What is exposome? But for those who are new, I just wanted to go ahead and get started with the definitions. So exposome is the totality of human exposures, whether it is exogenous or endogenous or both, all different types of exposures that we endure in our daily lives from conception onwards throughout the life course. So these exposures have an effect on health. That's what we are studying in exposome. Exposome complements genome, proteome, many other omics technologies. Exposomics is the study of exposome. And because I talked about how biomonitoring is integrated into exposome, I would say that biomonitoring is an approach, or biomonitoring is a tool to study exposome. And in fact, it is an important tool to study exposome. So biomonitoring is actually a direct measurement of people's exposure to toxic substances by measuring those substances or their metabolites in humans. So if we have blood or urine samples, we can assess the exposures. And this became popular about 20-25 years ago. When I was doing my PhD 35 years ago, we didn't know much about biomonitoring. And in the past 25-30 years, the analytical advancements have made these measurements at ultra trace levels possible in trace quantities of human specimens. And CDC considers biomonitoring as the most health relevant assessment of exposure. So with that said, I told you about how we can enhance biomonitoring, right? So I have 3 circles here. The central circle, the blue one, is actually the biomonitoring. We basically assess, use biomarkers of exposure by measuring chemicals in those, matrix for different types of chemicals, right? That is, sometimes people kind of look at it as a black box. You send the samples to a lab and they will give you the data. But there is a lot more to it, as I told you before. The biomonitoring can be enhanced by doing lots of different things, by thinking a little bit outside the black box. So when we make the measurement in urine and submit the data, the question is, oh, I have very high levels of, say monoethyl phthalate. The question is, where do I get exposures from, right? So with the same analytical methodologies, the instrumentation, we can be able to answer that by measuring chemicals in different exposure sources. So the green circle here on the left side is the exposure pathway analysis. So that can be done in conjunction with biomonitoring methods. The one on the right side, the orange circle, is monitoring the effect biomarker that can also be measured in urine or serum samples. That can make the biomonitoring much more robust. And you will see that at the end of the talk, how we integrate in our lab by combining biomonitoring, exposure pathway analysis, and including effect biomarkers in our urine or blood samples to make this program a fascinating research program in public health science. So when we talk about biomonitoring, often it involves analytical method development, right? We are measuring chemicals at ultra trace levels. It requires method development. And it's not as simple as people would normally think. Many chemists come to my lab, oh, give me the SOP, I will get the analysis done. But then they realize later that, we are measuring chemicals at trace levels, 1,000 to million times lower than what a clinical chemistry lab would be measuring for their biomarkers, cholesterol, triglycerides, red blood cells. Those are present in, serum samples at milligram or, several tens or hundreds of milligram quantities per milliliter of urine. But we are measuring chemicals at nanogram, picogram, sometimes even femtogram level. So that's biomonitoring basically. As I told you, if we just report the data back to the participants, the question is, where do I get the exposure from? So, by measuring chemicals in different sources of exposure, food, air, because we inhale air can be contaminated. We use a lot of personal care products, drinking water. So we measure chemicals in all those exposure sources to characterize the sources of exposure for a variety of chemicals that we measure in biomonitoring. That way we can tell, oh, if you are exposed to high levels of DEHP, it is very likely coming from your diet. Even within diet, what kind of diet can contribute to that exposure? That is important to put the biomonitoring data in the context of understanding the exposure pathway. When we communicate the results to the public, we need that kind of input. The effect biomarker. The question is, okay, if I'm exposed, are there some of the the effects that can occur because of that exposure? And that can be measured in urine also. So by combining biomonitoring with the effect biomarkers, you can also try to understand the disease outcome pathway, adverse outcome pathway. And we have done that. And I will present some of the examples of the work that we have done in the past 25 years, combining all these things. This is what basically my lab represents in terms of the research program we have here. We integrate chemistry with biology, environmental science, and of course, we work with epidemiologists, clinicians, toxicologists. So we work as a team. We learn from each other, from these kinds of approaches. So let's start with the first one. So here I have just taken the two circles. One, the biomonitoring, the blue circle I talked about, and the other one is the exposure pathway circle, the green one. So, 35 years ago when I started my PhD work, of course, I was doing exposure assessment for PCBs, PBDEs, pesticides. It was 35 years ago. At that time, my research was focused on measuring chemicals in food, water, air, and to calculate the exposure dose, right? Based on the amount you ingest for, different types of exposure sources and the concentrations of those chemicals you measure in those exposure sources, you can quantify the exposure dose, right? And that I call here as an external dose of exposure. And biomonitoring, basically measuring chemicals in urine, saliva, feces, breast milk, blood, toenails, all kinds of, biological specimens. And the exposures that you call or concentrations that you calculate from these biospecimens can be used to calculate the internal dose of exposure. So now we have exposures calculated from two different approaches, external dose, internal dose. Theoretically, they should match, right? So that's what we try to do in our research component. Do they match? If I am measuring phthalates in food, water, air, personal care products and calculate an exposure dose, does it match with the exposure dose calculated from by measuring phthalates in urine? If they don't match, then we are missing something. Maybe something wrong with the calculation, or we are missing some of the exposure sources. That's what we try to do. So dose is a function of concentration in blood or urine, or alternatively, you can say that concentration in blood or urine is a function of dose, because when you are dosed with few milligrams or micrograms of chemicals, some fraction gets into your urine. So the concentration in urine is a function of the dose that you ingest, right? So if external dose is equal to internal dose, then you can say that all the exposure sources are accounted for. But in many of our experiences over the past 25 years, what we have found was that the internal dose, that is the concentration that you measure in urine or serum, that is converted into a dose, is much higher than the concentrations you measure from food, water, personal care products, air, all kinds of exposures, which means we are still not accounting for some of the sources of exposure. The sources are not well characterized, right? That's what we did for many chemicals. And I'm going to show you an example with phthalates, because many of you know phthalates, widely studied chemical, thousands of publications out there. They are used as plasticizers. They are high production volume chemicals. And some of those phthalates are regulated in toys and childcare products, right? So many of you might know about dimethyl, diethyl, dibutyl, diisobutyl. These are all the low molecular weight phthalates, mostly used in personal care products and cosmetics. Fragrances are one of the important sources of diethyl phthalate. Diethyl xyl phthalate, benzyl butyl phthalate are mostly used in plastics, PVC type of plastics, vinyl flooring. They all contain diethyl xyl phthalate, and sometimes even much higher molecular weight phthalates are widely used in those kinds of plastics. And these are all reproductive toxicants. When you get exposed to phthalates, they get metabolized to monoesters. So diesters become monoester, and they get excreted in urine as glucuronide conjugates. Mostly urine, but we don't measure often feces. But I should say that phthalates are also excreted in feces, especially high molecular weight phthalates are excreted relatively at higher levels in feces. We don't measure feces, right? So sometimes if your exposure assessment is just focused on urine, you may be underestimating the phthalate exposure. So if you are looking for dimethyl phthalate, basically it gets converted into monomethyl phthalate and excreted in urine. Diethyl phthalate gets excreted into monoethyl phthalate. Diethyl xyl phthalate has multiple metabolites. We measure 5 of them. In fact, there are much more than 5 metabolites of diethyl phthalate is present in urine. I will show you some examples of that. So I told you we started our biomonitoring research almost 25 years ago. And as we were building the capacity and the capability around 2010, 2009, we built this phthalate metabolite method. And at the time, not much was known about exposure to phthalates in Asian countries. They didn't have any infrastructure to measure phthalates in urine. I have collaborations with scientists from many different countries. So at that time, we were interested in knowing kind of a baseline range finding study. It's not a full-fledged epidemiological studies with thousands of samples. It is just to know, are populations in other countries exposed to phthalates, right? That's kind of a simple basic question at the time. So we had samples collected from 7 Asian countries. Sample numbers range from 30 to 100 from each of those countries. Not much, but then it was at that time important to know if there was an exposure. So these are the range of concentrations we found. You can see the concentrations range widely. This is the sum of 12 phthalates we measured at the time. I kind of lumped it to make this presentation simple because I don't, I can talk more in detail about each of the papers that we published, but for this talk, I just wanted to give you a bigger picture of how biomonitoring can be enhanced and used in research programs. So of those 7 countries, you can see the samples from Kuwait stood out. The median concentration of sum of 12 phthalates is 1050 nanogram per milliliter. And concentrations as high as 19,000 nanogram per milliliter were found in samples from Kuwait. And then it was a surprise for us. We were curious to know what are the exposures. So our Kuwaiti colleagues, worked on phthalates. After that, they submitted some grant proposals to understand phthalate exposures in Kuwaiti population. And we found out that later, the Kuwaiti population uses a lot of fragrances in many different products, and monoethyl phthalate, predominantly diethyl phthalate, is widely used in fragrances, and monoethyl phthalate is one of the major phthalates found in urine from Kuwaiti population. So that's what we found in that particular study. So then I'm going to talk a little bit more about what does it mean, right? So at that time, 2007-8, the median phthalate concentration in the US population was 240 nanogram per milliliter. And the concentrations in many other Asian countries were somewhat similar or slightly lower. So when you say 240 nanogram per milliliter, what does it mean, right? Sometimes concentrations don't mean much, but there is a way to make it more useful. You convert the concentration into an exposure dose, right? That can be done using a simple pharmacokinetic approach. So if you know the concentration in urine, and of course we excrete approximately 2 liters of urine every day, we drink 2 liters of water, so approximately between 1.5 liter to 2 liters of urine excreted. So you can multiply that to get, nanogram or microgram of, the excretion every day. And of course, we use some of the other factors such as molecular weights and fraction of that chemical excreted in urine to calculate the daily intake or daily exposure dose. So with 240 nanogram per milliliter, that's the median concentration for the US population in 2007-2008. What is the dose? So using this formula, this is the dose that we came up with. So the US population at the time was exposed to 550 microgram of phthalates every day, which is 0.5 milligram. It's almost like everybody, eating 0.5 milligram of phthalates every day, right? That's the median value. Some may be exposed at much higher level. Then the question is, where does that exposure comes from? If I know that my urine concentration for some chemical is high, I will be very curious to know where I get the exposure from, so that I can do some intervention measures, lifestyle changes, so that I can reduce the exposure. So then we went after various sources of phthalate exposure. We published several papers during the time. In 2013, we published a paper on phthalates. Of course, at the time we also analyzed parabens, many other chemicals in perfumes, deodorants, nail polishes, and many other personal care products. We can find tens to thousands of microgram per gram of the product for various phthalates. So based on the amount of usage of those personal care products, for instance, in adult female, the exposure dose was calculated to be 22 microgram every day. And majority of the exposure from personal care products was diethyl phthalate. So if you have very high levels of diethyl phthalate in your urine, it is likely from the personal care products that you are using, likely from the fragrances that you are using, right? And of course, we have even more detailed information from the publication, what brand of fragrances have much higher levels. So we try to make sense out of the biomonitoring data by measuring the products that contain these chemicals. We measured these phthalates in indoor air, indoor dust. We went to many salons, nail salons, hair salons, schools, playgrounds, kindergartens, homes. we measured phthalate concentrations. We measured concentrations in indoor air, indoor dust. Based on the inhalation, we calculated the exposure dose. For instance, in adult female, it is 16 microgram per day from inhalation and dust ingestion. Okay, so 22 microgram from personal care products, 16 microgram from inhalation and dust ingestion, and then the diet, right? It's an important source for many chemicals. So we analyzed a wide range of food, like grains, cereals, oils, dairy products, vegetables, all kinds of things. And we measured phthalates. We can find few nanograms per gram to 1000 nanogram per gram. That was the highest concentration we found in food. So based on the ingestion rate of each kind of food, we calculated the daily exposure dose in adult female. It's 60 microgram per day. And in foods, DEHP is the major phthalate found in food. So if your urine has more of DEHP metabolites, it is likely coming from diet. It's not from personal care products, right? So our study at the time, we found out that the diet is the major source of exposure to DEHP, and personal care products are the major sources of DEP exposure in population. So then now you can bring the biomonitoring data and the external exposure data together. What did we find? So based on the urine concentration, I told you that there is 550 microgram of phthalate exposure every day. But the known sources like personal care products, indoor air and dust, and diet combined together account for only 98 microgram. So only 1/5 of the phthalate exposure is accounted for from the known sources. So there are many unknown sources. We have been studying phthalates for more than 20 years, but still there are many unknown exposure sources. In other words, the exposures are not well characterized, even for phthalates. So then we went after many other sources, right? Of course, in hospital settings, many medical devices, we have published papers on that. Many over-the-counter drugs, many pills contain phthalates. Phthalates are added as excipients in those pills, so we can be exposed. Many plastics, many fabrics, many clothes are made up of recycled plastics, and they can contain phthalates as well. Many products that we use in our daily lives, cell phone covers, we have found thousands and thousands of micrograms per gram of phthalates in cell phone covers. Organophosphate esters, thousands, percentage level. Every time you hold cell phone, you are exposed to phthalates, right? And of course, many sanitary napkins, sometimes we don't know that these products contain phthalates. So we go after, we try to characterize exposure sources of phthalates from those kinds of products. That's one way, you can enhance the research in exposure science. Then there is more than that. When we measure phthalate metabolites in urine, for instance, DEHP, as I told you, 5 metabolites, MEHP, MEHHP, MEOHP, MECPP, and MCMHP, right? 5 phthalate metabolites. When we tell the mass spec just to pick MEHP, it is supposed to pick one peak with that particular mass transition, right? We tell that MEHP as a typical mass fragments. We tell the mass spec, I want to pick that mass fragment because I want to measure MEHP. But then you can see the mass spec. This is MEHP, the red with the blue peak overlaid. That is the MEHP peak. But you can see there are so many uncharacterized, unidentified. We expect that they are phthalate metabolites, isomers, but we are not measuring them. Why? Because we are busy, we are preoccupied. We don't have time. I wish, these are the things the PhD students can go after. Maybe some of these biomarkers are more important than the metabolites that we are measuring, right? So we are missing a lot of information in our analysis because everybody is busy in the lab. If you ask anybody working in biomonitoring lab, they will say we are busy, right? But there are lots of opportunities for making discoveries. in the chemicals that we are already measuring for 20 years. So then next question, are we using the right matrix, right? When I talked about phthalate, I told, yeah, we are using urine, but phthalates are not excreted 100% in urine. There is some fraction excreted in feces. And for higher molecular weight phthalates, nonal phthalates, decyl phthalates, 10, 12, 20, 30 carbon phthalates are excreted predominantly in feces. So we miss those exposures if we are not measuring these chemicals in feces. So I want to give you an example with quaternary ammonium compounds. They are used as disinfectants in hand sanitizers, dishwash detergents, and many products. So recently we have done this study looking at quaternary ammonium compounds in urine. When you measure their metabolites in urine, concentrations are not so high. We were surprised. How is that possible? We use lots of disinfectants and sanitizers, and we are exposed to several milligrams of quaternary ammonium compounds every day just by using some of the products. But in urine, you don't find them. You find few micrograms. But then what happens to several milligrams that you are exposed to? Then we kind of started looking for these compounds in feces, and that's where you find them. 9,480 nanogram per gram, 9 microgram per gram, right? Of these quaternary ammonium compounds, many different types of quaternary ammonium compounds, they are in feces. So if we are not using the right matrix, you will be missing the exposure. That's what's happening in many of the biomonitoring studies. We are always busy, preoccupied. We tell sometimes, what our colleagues tell us to do. But if you, do kind of little bit more, a lot of opportunities for research. And are we using the right biomarker for exposure? So when I told you about phthalates, we are measuring 5 metabolites of DEHP, but there are more metabolites of DEHP that we are not measuring. And that's also true for many, many, many other chemicals that we are measuring. For instance, parabens, we are measuring parabens. They are used as antimicrobials in many products. And we normally measure, the parent compounds, parent parabens, methyl paraben, ethyl paraben, propyl paraben. But, in urine, only 2% of the parabens are excreted as parent compounds. They are mostly excreted as metabolites. Dihydroxybenzoate, it's a metabolite of parabens, many different parabens. It's not from one single paraben, but all the parabens get converted into 4-hydroxybenzoic acid or dihydroxybenzoic acid. If you are not measuring these metabolites, you are missing 95% of the quantitative exposure to paraben, right? People are not measuring metabolites. So we did a study in, again, 2013, about, more than 10 years ago. My postdocs often get excited about, some of the peaks that come out in urine. Dr. Kannan, we are measuring this, parent compound, but then there is some other peak that is, 100 times higher than this parent compound. Should we look for it? Should we characterize that peak? Yes, go ahead. And then, we spend time, and these are the things we find. If you are doing a real exposure assessment, we should pay attention to, lots of different metabolites. Sometimes it's easier said than done. Sometimes my students complain about, Dr. Kannan, you make things look so simple. I spent 2 years of my time and you explain that in 2 minutes as if it can be done in 2 minutes, right? But it takes time, but then there are lots of exciting opportunities for research in biomonitoring. That's what I wanted to say. So with that, I just want to say that the exposures are not well characterized, even for those chemicals that we have been studying for a long time. Also, the biomarkers that we analyze for those chemicals that we are measuring do not capture all of the exposures quantitatively. We are missing lots of the other biomarkers for those chemicals that we make the measurements for. And also the third component here is that there are lots of new things, right? Substitutes. So we are only measuring a fraction of the chemicals. 350 chemicals are biomonitored by the ENHANCE program, and many labs we follow, ENHANCE. So we develop capacity and capabilities for those chemicals, but then more than 40,000 chemicals are in the commerce, and several thousand chemicals are high production volume chemicals. And always there are substitutes, right? So there are, again, my message is that there are lots of opportunities for research. It's not just measuring and submitting the results to our colleagues. We can do lots of exciting things. So I want to give you some examples. So again, in 2010, 11, 12, we were also interested in other things, right? Substitutes. At the time, the BPA substitutes were not known. We were the first to report BPS, BPB, BPG, BPA, all kinds of BPs, right, in urine samples. So I just highlighted this slide here to show that in 2013, when we published this paper, so we were looking for BPA. The blue line is creatinine adjusted urine concentration. The red bar is the unadjusted urine concentration of BPA from, of course, 7 different countries, because at the time we were interested in exposures in other countries where they don't have capacity or capabilities. And I highlighted here Japan, because you can see in Japan, BPA levels were low. It's a developed country. You would expect that, that the concentrations can be similar to that in the US and other industrialized countries. But the levels were low. On the other hand, BPS levels were higher in urine from Japan and many other countries. Why? It's because in 2001, Japan regulated BPA usage in many products. But then they started using BPS in 2001. So you see the concentrations of BPS are much higher than BPA in urine at the time. So that was the first study. Nobody knew about BPS, BPF, BPZ, BPA, BPB, all kinds of things. We did those kinds of things, 12, 13 years ago. And we also characterized the sources of BPA. We know that canned food consumption is the major source. And but then at the time, only canned food was characterized to be the major source. But then there are other sources, currency bills and thermal receipt papers. Not much was known when we did that study 13 years ago. So when we measured the concentrations in thermal receipt papers, milligram per gram, parts per 1,000, right? Several milligrams, 5 milligram up to 15 milligram for every gram of that receipt paper. So every time you touch the thermal receipt paper, when you go to grocery stores from the cashier, or when you go to banks, the tellers will give you the receipt. It's all coated with the BPA. You touch them, you get exposed to BPA. And again, we published a paper at the time measuring BPS. In Japan, you can see that the BPS levels are higher, and they didn't have BPA in receipt paper. So what is found in receipt paper is reflected in urine, which means that the receipt papers are an important source of exposure to BPA. So when we published our study, Harvard University conducted some studies with the cashiers and the bank tellers. They did some intervention study to prove that receipt papers are an important source of exposure to BPA. So again, we did lots of multinational studies. For instance, paraben exposures are elevated in Korea 10 times higher than in exposures from other countries. It's because of the usage of cosmetics in Korea. It's much higher. So the paraben levels in urine are unique. When you do multinational studies, lots of interesting patterns develop. You find lots of interesting information because the diet, lifestyle, culture, religion, all those, many of those demographic factors can have an effect on your exposures, right? So those are the things that you discover when you do multinational studies. So in Korea, you have high exposure to parabens, even today. Chlorophenols, exposures are much higher in Japan than in many other countries. Chlorophenols come from chlorobenzenes, and chlorobenzenes are used in mothballs. And the mothballs are used widely in Japan. And if you go to closets, many of the closets in Japanese homes, they put mothballs so that there won't be any fungal growth or any other microbial growth, because mothballs are very good repellents of insects. Also, the humidity is very high in Japan. So, when easy for fungi to grow, right? So if you have mothballs, that takes care of the problem. So that's why you see very high levels of chlorophenols in urine from Japan. And pesticides, very high in Vietnam, right? When you do multinational study, Vietnam is an agricultural country. So chlorophenols, chlorpyrifos, malathion are some of the major pesticides that are widely used in Vietnam. You find much higher levels in those countries. I will stay there because my time is short. So I'm going to spend some time, maybe next 10 minutes, talking about how this exposure information can be used in exposome, right? How biomonitoring can be used in exposome. So this is a study. Are we using appropriate sample size for biomonitoring that can be used in exposome? So when we talk about appropriate sample size, normally, in epidemiological study, we calculate intra-class correlation coefficients. If one sample from an individual is enough to represent long-term exposure, right? You calculate the ICC value. You collect samples from an individual over time and you calculate ICC. If the ICC value is less than 4, then one sample is not enough. Then you can calculate how many samples you need, right? It can be done. If the ICC value is between 0.4 to 0.75, it's fair to moderate, representativeness of the sample. If the ICC value is more than 0.75, it's a good predictor of exposure. One sample probably reflects exposure. So when we did a study with VOCs, volatile organic compounds, you can see the ICC values from the spot urine is less than 0.4 for most of the metabolites, except one. And when we use the first morning void, early morning void, at least 3 or 4 moderate to excellent ICC values, but then again, most of them no, good representation, which means one sample is not enough. Then we calculated how many samples are needed. You need between 4 to 73 samples, depending upon the isomer. Some isomers need 73 samples to represent exposure of an individual for VOC exposure. Some, if you use 10%, 20% variability in your design, which means more flexibility, 18 samples for that compound needed to assess, to represent the exposure. So this is to calculate the individual variability, because every day our exposures vary, right? Depending on what you eat, where you were, what you were doing, all kinds of things, right? And also the product, depending on the chemical, the exposures can vary every day. However, when we do an exposomic study at the population level, there is population level variability that you should account for. So we have done these studies with our colleagues at Harvard University. Chirag Patel is a data scientist, bioinformaticist. We collaborate with them. We collaborate with epidemiologists, Germain Buck-Lewis. We worked with Germain for 10 years, providing analytical support for wide range of epidemiological studies. Thousands and thousands, tens of thousands of samples were analyzed for those studies. So in this particular study, we wanted to know, because exposome is dynamic, every day varies, the exposure varies, and there are lots of confounders, right? Covariates in your study design. So if you are going to study a semen quality from exposure to environmental chemicals, how many samples are needed? So for that study, we analyzed 128 endocrine disrupting chemicals in urine samples from the men who provided semen for that study. And then we also assessed semen quality parameters. So environmental chemicals and semen quality, right? This is what we try to do. Then are those 473 samples from men enough with all the covariates accounted for? The conclusion was that no, 473 samples are not going to provide confirmatory results of assessing exposure to the effects, right? So we found out that a sample size of 1,795 to 3,225 samples needed if we have to conduct an exposome-wide study to assess exposure to semen quality parameters. But if you look at most of the studies looking at chemical exposure and semen quality, the maximum sample size is 201. So you're not going to come up with a confirmatory study. right? You can produce, you can conduct lots of studies with a small sample size. They're not going to be helpful. So biomonitoring studies, working in conjunction with the data scientists and epidemiologists, we can make all these important information that will be useful in conducting exposomic studies. I'm going to skip some of the slides. We have done lots of studies related to exposures and those studies included in exposomic studies. What are the variables that we have to take into consideration, right? There is another study published in Environmental Science and Technology showing that the exposures are not captured well, even for a couple who are living in the same home, but they have different exposure. So, what are the variabilities in those exposures and how we can account for that variability? Those are the kinds of things that we have done and we have published, by, again, biomonitoring work, working with the data scientists and epidemiologists. Lots of information out there. I'm going to talk, maybe next couple of minutes about health effect biomarkers. So like exposure biomarkers, you can measure lots of health effect biomarkers: oxidative stress, nitrative stress, inflammation biomarkers, metabolomic disorder biomarkers. All those things can be measured in urine. Okay, we have developed methods for them. So for instance, one paper published last year, 2 years ago, we have 19 biomarkers of effect can be measured in one single extraction: oxidative stress, nitrative stress. So oxidative stress to DNA, RNA, proteins, fatty acids, also inflammation biomarkers. If you have any inflammation response from exposure, if you have metabolic disorders from exposure, those biomarkers can be measured. So if you measure exposure biomarkers to chemicals and exposure biomarkers to effects, then you can put them together to make sense out of it, right? So we have done studies with the measuring exposure effect biomarkers every day for, like 35 days, every day measure the effect, to see if the effect biomarkers vary on a daily basis. We have done studies looking at that. If somebody was doing something, say yesterday, I was barbecuing, I was eating barbecued food. I don't know if some of my oxidative stress levels elevated because I was doing that. Or yesterday, I was drinking some, wine. I don't know if some of my oxidative stress markers are elevated. Can you look at that? Or nitrative stress or inflammation, right? So you can do all those kinds of things by measuring effect biomarker. This is my one but last slide, so that I can finish on time. So what we are going from here, by doing everything together, we are trying to understand the adverse outcome pathway for human disease. Biomonitoring can be used to do that. This is something people are not doing that. In ecology, I used to do a lot of ecological work 20 years ago. We used to do those kinds of things, adverse outcome pathway for diseases by measuring exposures and then different stages, different biomarkers at intermediate levels of effect, right? So you have some exposure and you have an effect. And in between, there are lots of effect biomarkers that you can measure to link the exposure to effect. That will provide multiple lines of evidence to make the cause-effect linkage, right? Because cause-effect linkage in a population level study is difficult to achieve because we have so many variables. But then by providing multiple lines of evidence for an exposure to the effect, you can achieve that. You can accomplish that. That's what we can do in biomonitoring. That is very less studied, but this is the line, the direction that we are taking in our lab. We are trying to include hundreds of effect biomarkers in our urine, and then we have, we measure several hundreds of chemicals in urine. But then now we want to link that to make some sense out of exposure to effect linkage. So by saying that, I would like to conclude my talk by concluding that environmental chemical exposure is an important contributor to a variety of non-infectious diseases. I can say that having involved in this, working in this field for 30 years, environment plays an important role. Of course, I don't ignore genetics. Genes are important, but the environmental factors can be triggers for many of the dormant genes. If they are not expressed, you don't see the effect. Environment can trigger those genes. And environment by themselves can be an important factor in non-infectious diseases. And exposomics need to be expanded. We need to do more exposomic studies to understand how those factors, environmental factors, can contribute to disease. Biomonitoring studies permit discoveries. It's not just monitoring. It's not just providing service to our esteemed colleagues. We can do lots of exciting, fascinating discoveries. I am so excited. I wish I have, 100 hours every day. We can do lots of interesting research, right? It's a fascinating area of research in public health sciences. Biomonitoring also helps our colleagues, epidemiologists, toxicologists, clinicians to inform appropriate ways to quantify environmental, appropriate sample matrix, appropriate sample size, appropriate timing of sample collection, and many other factors that affect your data analysis that can be discovered through biomonitoring. We have done a lot of ICC calculations. What is the appropriate matrix for sample collection? What is the sample size? All those things can be done through biomonitoring methods, biomonitoring work. The effect biomarkers included with exposure biomarkers can help delineate adverse outcome pathways in identifying the etiology of the disease. That is exposure, effect, disease continuum. This is an area that has a lot of potential. We are already into it and we are making progress. And maybe next 2, 3, 4 decades, a lot more to do in this field. With that, I would like to acknowledge my funding agencies. CDC funded us for 25, 30 years. That's how we started this biomonitoring program. NICHD funded for 10 years. Again, we were providing support, analytical support for our NICHD colleagues. And then, last 7, 8 years, NIEHS funded through HERE program, CHEER program, and then HERE program to enhance our capabilities and capacities in biomonitoring. Thank you so much for this listening and appreciate your time. If you have any questions, I think I have time to answer those questions.
Thank you so much for such a wonderful talk, Dr. Kannan. I'm going to remind everybody of the ways that you can ask questions. You can either type them in the Q&A box, or if you want to be unmuted, you could raise your hand and we can call on you. We already have a question for you, and the question is, any unexpected exposure effects from the EWAS? How much of that data was usable despite the N limitation?
Yeah, the EWAS study at the time was focused on 470 plus men. It was life study, longitudinal investigation on fertility and environment. Dr. Jermaine Buck-Lewis was the principal investigator. So we had contract with NICHD at the time. We were the lab providing all the analytical support for a wide range of environmental chemicals. As I told you, we analyzed more than 128 endocrine disrupting chemicals in semen, urine, serum. And the finding was that the 400 plus samples were not adequate to make a firm or confirmatory conclusion from the study. We needed more than 1,300 samples at least to make a firm conclusion because of several covariates, confounding factors, that affect exposure levels in urine or semen at the time. But the data are out there. The semen quality, I think 45 different semen quality parameters were analyzed in addition to 128 endocrine disrupting chemicals in semen and urine from men at the time for the study.
Thank you. I actually had a question about early in the presentation, you talked about the sources of phthalates in the US, and you mainly just talked about female exposure. So I was curious how the male exposures compared and if there was differences like in the food versus the personal care products.
Yes, Jennifer, I used adult female just to keep my talk simple. We have done a lot of work. We have calculated exposures for children, males, females, teenagers, in all different age groups. That's how the exposure assessments are normally done. But because of the time constraints, I just wanted to give you an example of, how this science, the exposure science, can be done as biomonitoring as well as environmental monitoring. So yes, the adult females exposure through personal care products is much higher exposure dose compared to adult males. If it is 60 plus microgram per day from adult females, it's about half for adult males, right? So, but again, my reason rationale for just selecting adult female is to keep the talk simple so that you understand the concept of what we are getting here, what I'm trying to achieve here, in terms of the talk. It's again basically to compare the exposure doses from environmental exposure from biomonitoring to exposure, and what we are missing, and what needs to be done to find out the missing source, right, or the missing link here.
Thank you. So we have a couple more questions to get to. The next one is, are principal components analysis used in your studies?
Yes, of course. I'm not an expert. Of course, I do principal component analysis. It's not that complicated. But then our data scientists, we work with data scientists at NICHD. We have a team, biostatisticians, and at Harvard University, we have bioinformatics experts, big data scientists who have been helping us with all different way of analyzing the data. To take care of the confounding factors, covariates, all those kinds of things. Yes, principal component analysis were used in many of those studies to isolate some of the variables, and then lots of multilinear regression, bivariate all different kinds of analysis to take care of the covariates in those studies.
Okay, great, thank you. The next one is the person's thanking you for a great talk. And the question is, which mode of acquisition are you using in order to investigate whether other metabolites could be potential biomarkers?
Yeah, I mean, you can do lots of different ways, right? Discoveries, especially in analytical labs, these days with lots of instrumentation, Orbitrap, right? People use that, but unfortunately, Orbitrap, most of the work is qualitative unless you have the standards. You don't, confirm the chemical that you identified. Orbitrap, sometimes people say it's high resolution. Yes, it is high resolution, but high resolution doesn't mean high sensitivity. Resolution and sensitivities are different. When you use targeted analysis for phthalates, for example, right? So when you analyze metabolites of DEHP, 5 different metabolites I talked about, they're very sensitive. Much more sensitive than Orbitrap methods, high resolution mass spec methods. So when we tell the mass spec to look for a metabolite MECPP, as I showed you, there are many other peaks that come with the same fragmentation. So usually the chemicals fragment in the mass spec. That's what we look at. The parent ion and the daughter ion are very similar, are the same, similar to the metabolite that we are looking for, which means there are some isomers that we are not quantifying. We know that they are phthalate metabolites, but we don't know exactly the structure of those metabolites. They are maybe isomers because we don't have the standard, but we can say that they are phthalate metabolites. If we invest some time, if there is funding from some sources, Kannan, go after this metabolite, whatever you say. We have, maybe $100,000, do this characterization. Maybe we will, synthesize some standards, some isomers, and then we can quantify them, right? That's how it works in discovering some of those, unidentified compounds in our chromatograms, mass spectrum. It doesn't matter whether you use positive mode, negative mode. Yeah, if you have time, you can switch different modes and you can find out all these kinds of, interesting features in our chromatogram. And it is all about, going after them, how we approach them, how we, narrow it down to a chemical that we are looking for. We have done that for many different compounds. In fact, we have discovered many metabolites of benzothiazoles benzotriazoles in urine. We have published papers on that. I don't have time to talk about all. I can talk, maybe hours and hours of all those things we did. But there are ways to approach these kinds of issues and make discoveries in biomonitoring.
Okay, we have two more questions. I'm not sure if we have time for them, so I will try. Is there anything you've learned during your research or research consumption that have led to a personal lifestyle change in avoiding exposures?
It's not only to myself. I influence my family, my lab members. You can talk to my lab members what I tell them about. Of course, I mean this. It's a personal thing to me, honestly. I get my urine checked, once in a while. In fact, I got that checked 3 weeks ago doing a multi-panel novel chemical method that we developed recently. I was surprised. I had 3 ppb of monoethyl paraben. Of course, it is 50 times lower than my colleague in the lab. That is kind of, okay, I'm not too bad. But then where did I get the 3 ppb, right? Immediately makes me wonder. Of course, parabens are used in many processed food, right? If you eat cake, any food that's on the counter in grocery stores, they add paraben because antimicrobial property, right? Prevent mold growing from you on your cake. So if you do that, you can get exposed. So yes, I take these measurements very personally, not only to myself, but of course I talk to my wife, my children. They are very probably influenced by the work that I do. Of course, talk to my lab members, and of course, they are more interested in what they are exposed to. I'm passionate about what I'm doing, and of course, that is infectious, right? When you work with people like me, my lab members care about what they're.
I think we're all out of time. We did have one more question, and we can get that to you, Dr. Kannan, and maybe you can respond to the person asking the question. So thank you so much for your excellent presentation. I also want to take a second to extend a special thank you to the organizers of this series, as well as the HERE Coordinating Center for their logistical support today. And also thank you to all of you for your attendance, attention, and engagement in today's event. To learn more about upcoming webinars or to access recordings from previous webinars in this series, please visit the HERE program website at hereprogram.org.